Welcome to Chem Help ASAP. In this video, we'll see an interesting example of an enol tautomer that is more stable than its keto form. The synthetic work in this video was performed by the research group of Professor George O'Doherty of Northeastern University in Boston, with biological screening by the group of Professor Deborah Lannigan at Vanderbilt University in Nashville, Tennessee. This research was published in the Journal of Organic Chemistry. The citation is on the following slides as well as in the video description. Across the top of the screen are the keto and enol tautomers for cyclohexanone. For cyclohexanone, the keto form is strongly favored by a factor of about 1,000 to 1. At the bottom of the slide is another cyclic ketone with its enol tautomer. Interestingly, this molecule, unlike most, strongly favors the enol tautomer and not the keto form. Why would this be? This surprising observation may be explained in a couple ways. First reason, by looking at resonance forms of the enol tautomer, starting from the ring oxygen, if we push the electrons around the right side ring, we can draw a resonance form that generates a 10 electron aromatic system that includes both rings. 5 pi bonds for 10 electrons to satisfy the 4n+2 rule of Huckel's rules. Second reason, an intramolecular hydrogen bond likely also helps stabilize the enol form. So in order to gain stability from aromaticity and hydrogen bonding, the molecule favors the enol form over the keto form. The dominance of the enol form also affects the reactivity of molecules like these on the bottom of the slide. The O'Doherty group treated a related molecule to the far left with a weak base, potassium carbonate, and a primary halide. The base deprotonated the enol to form an enolate. Both resonances of the enolate are shown. We are accustomed to seeing enolates get alkylated on carbon, as shown on the bottom. In this case, however, perhaps for reasons we have discussed, or perhaps because of steric hindrance from this aromatic ring, this enolate is preferentially alkylated on oxygen and forms a new ether. The enolate undergoes O-ALKYLATION instead of C-ALKYLATION. The O'Doherty group made the previous molecule and structures like it and removed the 3 benzyl groups through hydrogenolysis. The final products were tested by the Lannigan group and showed moderate activity against both an enzyme associated with certain forms of cancer and a tumor cell line. In conclusion, research groups at Northeastern and Vanderbilt worked with an interesting series of molecules that favor the enol tautomer over the keto form and exploited the reactivity of these molecules to research new potential anti-tumor compounds. If you are interested in learning more about the work of these two groups, then check out the reference on this slide or in the video description. Please subscribe, like, or leave a comment. Take care.